Welcome to the deep dive. This is where we take some key sources, sometimes just one really fascinating study, and break it all down for you so you walk away really understanding what's going on. Today, we're tackling a pretty critical ecological puzzle. How do you figure out who's eating whom in a sensitive place like a pond without, messing everything up?
It's a classic problem in ecology.
Exactly. And we're diving into a 2023 paper from Oecologia. It's called Using eDNA to Understand Predator-Prey Interactions Influenced by Invasive Species, led by Maria Riaz and colleagues.
Right.
Our mission, really, is to unpack how they use some cutting-edge genetic tech to solve this old mystery, specifically proving how invasive species impact vulnerable native animals.
And this isn't just academic, is it? There's a huge urgency here. Amphibians globally are, well, they're in crisis.
I can hear about it all the time.
Yeah, the decline rate is frankly shocking, something like 200 times higher than historical rates, especially since the 80s. And when you look for the main culprits, invasive predatory fish are often right at the top of the list.
Which makes intuitive sense, right? Because amphibians, they spend that really vulnerable larval stage in the water.
Completely exposed.
Yeah, basically sitting ducks for any aggressive newcomer fish that decides to set up shop in their pond.
Precisely. But the tricky part, traditionally, has always been proving that connection. Demonstrating that the fish are causing the decline across a whole population.
How did they used to do it? Netting? Traps?
Things like that, yeah. Netting, electrofishing, traps. But these methods, they can be really invasive. You might disturb the habitat, you might accidentally harm the animals you're trying to study, or you might just miss the rare species altogether.
Okay, so that sets the stage perfectly for environmental DNA, eDNA.
The game changer.
It sounds like the conservationists', secret weapon. Instead of catching the animal, you just scoop up some water and look for the DNA they leave behind. Skin cells, waste.
That's the essence of it. It's wonderfully non-invasive, and the sensitivity is just remarkable. Oh, yeah. eDNA can pick up species that are there in such tiny numbers or are so elusive that your traditional surveys would just completely miss them.
Okay, so the Riaz study used this eDNA, but they weren't just trying to say, yep, fish are here, amphibians are here too, right? There was more to it.
Much more. They had a couple of really key goals. First, they wanted to test if eDNA could work quantitatively, not just presence-absence, but measuring how much DNA.
To see if it could actually reveal active predator-prey dynamics?
Exactly. Could they show, for instance, that where you find more predator DNA, you find less prey DNA? That kind of negative correlation.
Okay. And the second goal?
The second was to use this method to really try and confirm the hypothesis for their specific study area, that the local amphibian declines they were seeing were, in fact, linked to these invasive fish moving in.
Got it. So where did they do this? You mentioned a specific area.
Right. They chose a place called Wettwer in Hessen, Germany. And this is important. It's a protected area, part of the Natura 2000 network.
So supposed to be a safe haven.
Supposedly. Yeah. And historically, it was known as a real amphibian hotspot, which makes the findings even more, well, concerning.
It really underscores the threat, even in protected zones.
It does. So within this roughly 70 square kilometer area of wetlands and ponds, they focused on 4 key species.
The players in the drama.
You could say that. 2 potential prey species, the amphibians. First, the garlic toad, which they called A1. It's actually critically endangered locally in Hessen.
Wow, okay.
And second, the crested newt, A2, which is also protected under EU law. So 2 species already under pressure.
And the predators, the invasive fish.
2 of them, both listed by the EU as invasive species of particular concern. You've got the sunfish, Lepomis gibosus, that's F1.
Okay.
And the one that features really heavily in the results, the topmouth gudgeon, Pseudoresbora parva, that's F2. Apparently, it has a real taste for amphibian larvae.
Okay, so we have A1 and A2 are vulnerable natives versus F1 and F2, the invasive predators, in this protected German wetland. How did the researchers actually gather the data?
They ran a pretty intensive sampling campaign. It was during the amphibian breeding season in 2018, May, June, and July.
Over 3 months.
Crucially, yes, over time. They took water samples from 89 different spots across 31 ponds. That temporal aspect, watching things change month by month, is really key to their analysis.
And when they got those water samples back to the lab, it wasn't just a yes or no for DNA. You mentioned quantification.
Exactly. That's the technical step up. They used quantitative qPCR. Think of it like PCR, which copies DNA. But this method measures how much DNA you started with.
How does that work?
They use specific genetic markers called Tachman assays designed to only find the DNA of their target species, A1, A2, F1, F2. They filter the water, get the eDNA, and the qPCR machine amplifies it. The more cycles it takes to detect the DNA, the less there was to begin with. Conversely, quick detection means a higher initial concentration.
Okay, but let's clarify that. A high concentration of, say, top mouth gudgeon DNA, that doesn't mean you can say there are exactly 50 fish in this pond, does it?
No, absolutely not. That's a really important point. eDNA concentration isn't a direct count of individuals. It's more like a proxy for relative abundance or biomass.
Right.
So a pond with lots of fish, or bigger fish, will likely shed more DNA and have a higher concentration in the water sample than a pond with just one or two small ones.
But for seeing patterns, like if high fish presence correlates with low amphibian presence, that relative measure is enough.
It's robust enough for these kinds of ecological correlation studies, yes. It tells you who's sort of dominating the genetic signal in the water.
Okay, that makes sense. So they had the qPCR data. Did they do anything else to check it?
They did. They ran traditional surveys alongside the eDNA, sampling things like using traps for the fish and listening for the garlic toad's calls, mainly to confirm the basic presence or absence detected by eDNA. Cross-validation.
Good practice. But the really clever part was the statistics, wasn't it? Something about Monte Carlo tests and transitions.
Yes, that was the analytical heart of it. Instead of just looking at a snapshot in July and saying, okay, A1 is absent here, F2 is present here, they looked at the changes between months.
Changes, like species moving in or out.
Exactly. They analyzed the transitions, May to June and June to July. For each site they asked, did amphibian A1 enter this site? Did it persist for the previous month? Or did it disappear?
Ah, okay.
And then, crucially, they used Monte Carlo simulations, basically, a way of testing if observed patterns are statistically significant or just random chance to see if those amphibian transitions were linked to the transitions of the predator fish at the same sites over the same periods.
So it's not just are they coexisting, it's is the arrival of the predator linked to the disappearance of the prey? Or does the prey only show up where the predator isn't?
Precisely. It moves beyond simple co-occurrence to look at the dynamics of invasion and exclusion, or avoidance, playing out over time.
Why is that transition analysis so much more powerful for showing a predator-prey interaction?
Because it captures the ecological action. Like you said, if you just check in July and find fish, but no amphibians, maybe the amphibians were never there. But if you see the amphibians disappear between May and June, and that disappearance happens significantly more often in ponds where the invasive fish just arrived, well, that's much stronger evidence.
Right. It points much more directly towards cause and effect, even if it's still technically a correlation.
It provides a much stronger correlational link, yeah.
Yeah.
It's showing exclusion happening.
Okay, so moment of truth. When they ran these Monte Carlo tests on the transition data, what did they find? Was there a clear link?
Oh, absolutely. The results were pretty stark. They found a significant negative relationship between the eDNA concentrations of the invasive fish and the native amphibians.
Meaning more fish DNA generally meant less amphibian DNA.
That's the overall pattern. But the transition analysis gave specifics. For example, the entry of the garlic toad, that's A1, the critically endangered one, into a new pond, was strongly, significantly correlated with the absence of the top mouth gudgeon F2.
So the toads were basically only colonizing ponds that were free of that specific predator. Avoidance.
Looks like it. They were either actively choosing predator-free zones or perhaps only successfully establishing where F2 wasn't present.
What about the other way around? Did they see the fish moving into amphibian territory?
Yes. The top-mouth gudgeon, F2, showed that negative relationship with both amphibian species, A1 and A2, especially between June and July, and perhaps most tellingly.
Yeah.
They documented F2, the gudgeon, successfully invading ponds, where one or both of the amphibian prey species were already present and persisting.
Wow. So the predators were actively moving into areas where the prey was established.
It strongly suggests that, doesn't it? Like
Yep.
the presence of the amphibian larvae might even be attracting the fish.
And didn't the paper mention this lines up with older, more direct evidence?
It does. They mentioned that previous studies in the same area had actually looked at the stomach contents of captured topmouth gudgeon, and they found larvae of both the garlic toad and the crested newt inside the fish. So direct evidence of predation exists.
Right. So the eDNA work isn't discovering the predation itself for the first time, but it's showing the ecological consequence of that predation at a population level across many sites non-invasively.
Exactly. That's the power. It confirms the impact across the landscape quickly and without disturbing anything further. It links the known predation to the observed population dynamics.
So pulling this together, what are the big takeaways here? For conservation, this seems pretty important.
It's huge. The study basically provides strong empirical backing for the hypothesis that the local declines of these protected amphibians are, at least partly, maybe significantly, caused by these invasive fish spreading.
It takes it from suspicion to, well, data-driven evidence.
Right. For people managing that land, the Auenverbund Vettero, it means they now know that protecting those amphibians effectively must involve dealing with the topmouth gudgeon and the sunfish. It helps target conservation efforts.
So eDNA becomes a critical diagnostic tool.
Definitely. Gives insights into these hidden drivers of decline and should really be part of the toolkit for monitoring biodiversity loss.
Now, we should also be critical here. As powerful as this is, it's still based on correlations, right? eDNA concentrations linked to presence-absence transitions.
Absolutely. The authors acknowledge this. Correlation doesn't equal causation, that's the classic mantra. While the link is very strong and statistically significant, you can't definitively rule out all other factors.
Like what? What else could be going on?
Well, the study period was May to July. That's relatively short. What if there was, say, an unusual heat wave or drought during that time?
Which we know hits amphibians hard anyway.
Exactly. Environmental factors like temperature and water availability definitely affect amphibian breeding success and survival. It's possible those factors were also contributing to declines, maybe even interacting with the predation pressure.
So could the fish just be thriving in ponds that were already stressed environmentally and the amphibians were declining anyway?
It's a possibility that's hard to completely disentangle with just 3 months of data. That short window is a limitation they mention. However,
Yes.
the consistency of the negative relationship across many different sites and the specific pattern of amphibians disappearing when fish arrive still points very strongly towards predation being a major direct driver. It's the most immediate explanation the transition data supports.
Okay, so it's strong evidence acknowledging potential complexities. Where does this research lead next? What's the future for using eDNA like this?
Well, the obvious next step is scaling up. This study was elegant, but focused on just 4 species. The real power comes when you apply this to whole communities.
How do you do that? Run hundreds of qPCR tests.
You could, but it gets cumbersome.
Okay.
The next wave is using techniques like eDNA metabarcoding.
Metabarcoding.
Yeah, instead of species-specific tests, you use universal primers that amplify a specific gene region like a barcode from all fish or all amphibians or even broader groups in the sample. Then you sequence everything that gets amplified.
And that tells you?
It gives you a list of potentially dozens or hundreds of species present in that single water sample. You can start to map out entire food webs, see how multiple invasive species interact, or how the whole community shifts when one key player arrives or disappears.
So moving from simple predator-prey pairs to complex community interactions.
That's the goal, using eDNA to get a much richer, system-level understanding of these biotic interactions.
That sounds incredibly powerful. So wrapping up this deep dive, it really hits home that even in areas we designate as protected, like that German wetland invasive species, can just silently creep in and really destabilize things.
It's a constant battle, unfortunately.
And this study is a brilliant example of how a non-invasive tool like eDNA gives us the speed and the insight to actually diagnose these hidden threats before it's potentially too late.
Early detection and understanding the dynamics are key.
So maybe a final thought for you listening. Think about your own local environment. Could be a park pond, a stream, a nature reserve. Are there native species struggling? Could there be hidden invaders? Tools like eDNA offer a way to start asking those questions and maybe get answers much faster than we ever could before.